Now we're going to take a look at some other techniques for genetic engineering. There are 4 that we're going to take a look at. Some of these terms are one of them probably isn't new to you, the rest probably are. We're going to look at gel electrophoresis, PCR, DNA fingerprinting, and cloning. Okay, when 2 of these go together, and we'll look at those together, that would be gel electrophoresis and DNA fingerprinting. Let's take a look at PCR. PCR is a technique that is called polymerase chain reaction, and it creates multiple, and we're talking millions, of copies of the same piece of DNA. The simple way to show you, this diagram maybe doesn't make a whole lot of sense, but let me show you. Here's a piece of DNA. Whoops, sorry about that. I'll call up my ink a second. There's a piece of DNA right here, and we want to copy that DNA. Well, you can have the cell do it, but the easiest way to do it, to make it copy, is you put the DNA into a vat, into a test tube, and you heat it. When you heat DNA, the strands split apart. Okay, so here's one side of the DNA and here's the other. The strands split apart. So now what you need is you need to add pieces of DNA. Well, you need to add literally A's, T's, G's, and C's into the test tube. And the interesting thing is you can get A's, T's, G's, and C's just about anywhere. Every living thing has DNA. And so chemical supply stores have adenine, thymine, guanine, and cytosine bases. So you buy them, you dump them into the test tube, and now you need, it says TAC-Pol here, you need to add DNA polymerase. DNA polymerase is the enzyme that DNA uses when it copies itself to put itself together. So I'm going to draw this up on the left-hand side, or up on the right-hand side here in white. So you add those new pieces of DNA. They come in and get attached. And you have 2 new pieces now. Where you had 4, where you had 1 copy of DNA, you now have 2. Well, then you heat it again. Do the same thing, and you go from 2 to 4. Heat it again. Do the same thing, you go from 4 to 8. Keep doing that. If you go for 20 more cycles, you got 2 million copies of the same piece of DNA. Okay? Why would you want to do that? Well, as we'll see, let's say you have very little DNA. Let's say if you watch CSI or some of those shows that they collect DNA from a crime scene or from a person and they don't have a lot of it, you can take that same piece of DNA and make millions of copies of the exact same one. Okay? It's called PCR. The second thing we're going to talk about is something called gel electrophoresis. This seems very complicated. It's actually kind of a simple process. There's a diagram of it, and I'm going to explain this it can be a little bit confusing, but it will help you understand how we can identify criminals or do parental tests or whatever. Okay, in gel electrophoresis, it is used to separate pieces of DNA that have been first cut by restriction enzymes. Okay, so let me explain this by taking here's piece 1 of DNA, and here's piece 2. And let's say this piece of DNA came from me, and this piece of DNA came from you. Now, those 2 pieces of DNA are different because you and I are different people, so we should have different DNA. So if we put the same restriction enzyme to cut my DNA, let's say the restriction enzyme cuts mine there, there, and there. Question to ask yourself right now, is it going to cut yours in the same place? Well, if yours is different than mine, it should not, right? Because restriction enzymes recognize specific sequences like AATT. So you're going to have your AATT in different places, maybe there, maybe there, maybe there, maybe there. So for the same piece of DNA from the same chromosome, you and I have different what we call restriction sites. Okay, so what? Well, now what we do is we take that, so we did this to my DNA in a test tube, and we did this to your DNA in a test tube. So now your DNA is all in little pieces. Your DNA should be in all these little pieces in a test tube. There should be 1, 2, 3, 4, 5 pieces. My DNA should be in 1, 2, 3, 4 different pieces in a test tube. And so, if we take my DNA and put it into, okay, we build what's called a gel. And in class, we'll show you what a gel electrophoresis apparatus looks like and show you what a gel would be like. So we're going to load these into something called a gel. And it literally looks like a piece of Jell-O with little holes in the end. We put those into the gel. And let's say we put mine in the first one and yours in the last one. Okay, and I'm going to ignore all these right down here for right now and just use the top here a second. So yours is loaded in blue. Mine is over here in red. And remember, mine was in 4 pieces and yours was in 5. When DNA happens to have a negative charge, negative. So that means if we run electricity through this, and this side is the positive side, and this side is the negative side with batteries, and again, we'll show you what this looks like, that the DNA would be pulled, because DNA is negative, it'll be pulled towards the positive pole. So the DNA is pulled down through the gel. And as it does, it separates. And mine, because I made 4 pieces, will have 1, 2, 4 bands. Yours, because it had 5 pieces, will separate into 5 bands. Because remember, when I cut up your DNA, for instance, we had something like this with 5 pieces. And so this piece, I'll label piece 3, travels to there because it's longer than, say, piece 1, which is down here at the bottom, because it's shorter. And they have to be pulled through the gel. That's not an easy concept, but I think you don't necessarily need to know the concept as much as what happens when we're done. So we can tell the difference between my DNA and your DNA by running them on this thing called a gel. When we do that, that's useful for many applications. Number one of which is something called DNA fingerprinting. Now, people get confused with DNA fingerprinting because they think it has something to do with your fingers. And this really should be DNA identification. Using DNA to identify somebody or something. So, why do you and I have different parts of DNA? If you think about it, for most of the stuff that we have, we're exactly the same. We have a 4-chambered heart. We have 2 lungs. We have 2 arms. We have 2 legs. We have 10 fingers and 10 toes. We have a nose with 2 nostrils, 2 ears, etc., etc., etc. There are very few pieces of our DNA that are not the same. But one important idea is that much of your DNA has no meaning at all. It's either old useless genes or repeated stretches of the same code. AG, 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 AG for thousands of bases. We don't know why this is. We'll call it junk DNA. It really serves no use as far as we can tell. The interesting thing about you and me is that even though our genes might be very, very similar, like for example, me and my brother would have very similar DNA, but my DNA still has different amounts of this junk DNA between our genes. So, how do we do DNA identification? Well, again, we use a restriction enzyme. We use gel electrophoresis. And when we do that, we'll get a pattern on a gel like we just talked about. And as far as we can tell, this is accurate to 1 in a billion people. On the right is a quick example of a paternity test. A paternity test, remember, is, and maybe you've seen this on Jerry Springer or whatever show, Montel, or I don't know what morning shows are on anymore, or shows that are on that people want to find out who the daddy is. Okay? So, if we want to know who the daddy is, we check the DNA. So here's a gel electrophoresis. This is the mother. Mom's DNA made 2 bands on the gel, and they've simplified this because a real one would have hundreds. Here's the first child's DNA, and notice that the first child's bands match one of mom's. Why does that make sense? Well, you have 50% of your parents' DNA. Half of your mom's and half of your dad's. And let me highlight all the ones where the kids are the same as the mom. I'll do them in blue. Child 2, there. Child 3, there. Child 4, there. Each of the kids matches mom in one spot. Now I'm going to put in a different color the ones that don't match mom, which you've already probably figured out, and you're thinking this is a waste of my time. Okay? This one for this kid doesn't match mom. This one for this kid doesn't match mom. This one for this kid doesn't match mom. That one for child 4 doesn't match mom. So let's say they're trying to determine who the father is. And they have this guy come in and he does a DNA test and check it out. Here are his 2 bands. And notice that these 2 kids match him there, and these 2 kids match him there. Voila, you're the daddy. Okay, he's the daddy. If another guy was tested, and his bands were like here, it would be obvious he doesn't match. He's not the father. That's DNA identification. In the same way, you can do it for crime scene DNA, except in a crime scene, you'd look for an exact match. Our last genetic engineering principle is cloning. Many of you have probably heard of cloning. It was thought to be impossible until 1997, when Dolly the sheep was cloned. Okay? By a guy named Ian Wilmot. In 1997, he cloned a sheep. Now, the concept of cloning is pretty simple. You take a single cell from an organism and all of its DNA, and you use that to make a genetically identical individual. And this should say, so I'm going to, and there's not really notes on this as much as there is to just take a look at what happens, what the idea is. You take a sheep and you take a cell from that sheep. This is the sheep we want to clone. This sheep is going to be the surrogate mother. Because if you want to make a clone, you have to have a mother for it. You take the cell and you take its DNA from this sheep. You take an egg cell from the mother, unfertilized egg, and you take out its DNA. Why would you do that? Because you don't want any of this DNA in there. You want all the clone's DNA. So you take out the DNA from the surrogate. This says fuse cells. There's a couple of different ways you can do it. You can fuse the cells, or what they do nowadays is they take a needle and they take all the DNA out of here and they inject it into there. So now what you have is an egg cell with a complete set of DNA. If this were humans, it would be all 46 chromosomes. Right? Be all 46 chromosomes. Now here's the problem. What they couldn't figure out is how to get this cell to divide. Because in real parenting, when sperm meets egg, that's the signal for the egg cell to start to divide eventually. But here we couldn't figure out, they could not figure out for a long time how to do it, and they experimented with a lot of different ways. And what they finally realized, and I'm going to erase the ink here and write this in, is that one way to get the egg to divide is to shock it with a little bit of electricity. An electric shock. And the egg cell starts to divide. So now you're dividing, now you're doing mitosis and getting copies of all the chromosomes. So you take that, now that cell is divided into 2 or 4 or 8, and you stick it into the surrogate mother. And I don't know why they drew the surrogate mother a different color than that one, because you'd want to use the same sheep as the mother. And she carries it to term, has a baby, and that baby then is a clone. And now, and by 2012 now, we've cloned many different kinds of animals. This, I think, has a lot of promise for treatment of disease. If we can figure out how to clone organs. Okay? Or if I could have a clone of me, then if I ever needed a transplant of any kind, like a kidney transplant, I'd know where to go. Because my clone has the exact same DNA. Everything would be a match. Okay, that doesn't mean maybe it does mean I'm making a baby just for me. There are a few problems with cloning that have cropped up. One of them is that only about 1 in every 1,000 clones for mammals, maybe 10,000 depending on the mammal, makes a successful clone. It's pretty rare that you get a successful clone, so you kill a lot of embryos. Now, for dogs and cats and sheep, that might not be a big deal, but for humans, it would be a really big deal, I think. Second, for some reason, clones, and we don't really know why, clones seem to have more problems than normal organisms. In other words, normally conceived organisms, babies, have less problems. Clones seem to develop genetic diseases and cancers at a much higher rate than normal organisms. But if you wanted to know how cloning works, that's how it works. Okay, and there are problems with cloning, which is one of the reasons we're going to look at, we're going to have certain people look at cloning in a little more detail with a project coming up. That is the 4 techniques of genetic engineering we use today now.